Hello guys, so today we're going to be studying about DNA and RNA, so hopefully you remember these things. So DNA, this is the deoxyribonucleic acid. The structure of this is a polymer made by nucleotides, and if you remember, a nucleotide is made by a phosphate group, pentose or a sugar molecule, and a nitrogenous base. Then in the nitrogenous base of DNA, we have two types of nitrogenous bases. We have purines and pyrimidines. In the group of purines, we have adenine and guanine, and the pyrimidine, we have thymine and cytosine. So if you see here, the pyrimidines, they have just one ring, and cytosine, uracil, and thymine, they just have one ring. Notice here that we have uracil here that is found in RNA, not in DNA. So that's the difference between DNA and RNA. So then the purine, they have two rings, as you see right here. Adenine and guanine, they have two rings as well. So the phosphodiester bond, this is the bond between the pentose, the sugar, and the phosphate group. So right here we have the phosphodiester bond, or right here we have the phosphodiester bond. So this bond is more stable than hydrogen bonds. The hydrogen bonds, if you remember, we find them between nitrogenous bases. So right here, as you see. So this is the structure of the DNA. There are two long nucleotide chains that are twisted about one another. This is a twisted letter or double spiral structure, or we can call it as a double helix. So again, the pentose or sugar, the phosphate group, and the nitrogenous bases, and between nitrogenous bases we have the hydrogen bonds. So the base pairing, always remember that we're going to have adenine in front of thymine with two hydrogen bonds, and guanine in front of cytosine with three hydrogen bonds. The ends of DNA strand are called 5' end, and then this ends at the phosphate, and the 3' end at the deoxyribose end. So the bond between nitrogenous bases is less stronger than the bond between the phosphate group and the sugar. And this property is very important because it will allow DNA to separate without damaging DNA during the synthesis of a new molecule of DNA. So as you see here, we have different sequence of nitrogenous bases that will make different type of amino acids if we are working on making proteins. So the genes in this, the nucleotides of DNA can be displayed in any order. So each sequence will represent a unique set of instructions with the information for the synthesis of a polypeptide chain. So genes, there is a specific sequence of genes that codify a final product of proteins. The unit of genetic information is the gene and consists of a portion of DNA molecule that controls a particular trait. It could be the color, the hair color, the eye color, the function of the cells of our heart, or other functions. So how people determine the presence of genes in DNA? So at the beginning of the 20th century, there were people knew that the chromosomes are in the genes and made by DNA and proteins. And through different experiments, they could prove that the genes are in DNA and not in the proteins. Before, they didn't know where the genes were, or in DNA or in proteins. So in 1928, Frederick Griffith did an experiment, and in his experiment, he used two strains of bacteria, two cepas. So the strain of bacteria was the Streptococcus pneumonia. The Streptococcus pneumonia S was a strain that was virulent, so it means that this bacteria produce illness. But then he used the strain R, and we need to remember that the strain R didn't produce illness because it was a non-virulent bacteria. So the characteristics of the the S strain was virulent, and it produced smooth colonies, and produce a polypeptide um, capsule that makes this bacteria virulent. And then we have the living R strain or R strain. The colonies by this were rough, no virulent, and it didn't have this capsule of carbohydrate. So in the first experiment, in the first mouse, he inject the S strain into the mouse, and the mouse dies. Then he took a sample of the blood and he could observe this bacteria, S strain bacteria. Then in the second one, the mouse injected with the R strain that was not virulent. In this case, this mouse lives. In the third case, what happens was that Griffith hit and killed the bacteria, the S strain of the bacteria, and then he put this injected into the mouse, and then the mouse lives. So the bacteria didn't kill the mouse. And in the last experiment, he uses the living R strain that was used here, and then the dead S strain that was used here. When he did this mix and injected into the mouse dies. With this, he said that something happened with the bacteria because when he took a sample of the blood of this mouse, he obtained living S strain. With that, he could say that the S strain of the bacteria modified something because of the S. So the R strain wasn't R strain anymore and it became S strain. So with this, he said the genetic material was in charge of sending signals, receiving and passing signals to the next generation. So with that, we can see that something was modified in the R strain to become S strain. And with that, we have the transforming principle.